Hi Hires, DNA in the Genome unit. We're looking at gene expression. Okay, so the first thing to be clear on is the difference between these two terms. And you saw them in that 5. So the genotype is all the alleles that you have in every single gene locus in your genome. Okay, so every particular gene that you have, what types of alleles are present. The phenotype is what you then see. So what that really means is how these genes are expressed. And by expressed, we mean that they've been copied from the DNA, translated into a protein that can then do a job. Okay. It's obviously not that simple, because why would we make it that simple? So first of all, you can pick and choose exactly what's going to happen with that gene expression, and it could be by signals that are coming from inside the cell, or it could be from signals outside the cell. So an obvious outside signal might be a hormone. A hormone triggers a receptor and then makes particular things happen inside cells. But it could be things like you're running out of a particular chemical and that triggers the production of a protein that can then synthesize it from something else. So you have all sorts of potential signals that are going to alter what your DNA is doing. Okay, then you have the possibility that different alleles will interact to give you a range of phenotypes. Now that, you should have heard the term polygenic. So that basically just means a trait which has many genes, okay, as opposed to monogenic, where you have a single gene that's deciding a particular expression. So you can also decide how long you switch a gene on for, how much of it you produce. And then on top of that, the proteins that you make can then be altered as well. So it's really, really complicated. But then that's not surprising, because we were talking about how you actually make a complex system work, and a cell is a wonderfully complex system. And then on top of that, we're going to see the environment can also have an impact. So what we mean by gene expression, we need to take somehow the information you've got on your DNA and make that into something that can then do a job. So to do that, you have two processes: transcription and translation. Okay, so transcription, if you order a transcript of your university course, what they'll give you is a list of all the things that you did with all of the course content that you had, the grades that you got. That's what a transcript is. If you watch any of these programs that are police procedurals or court procedurals, you might hear them saying that it's a court transcript. It's a copy of what happened. Okay. The translation is obviously shifting from one language to another, because that's what translation is. So the code that we have is DNA code. So that's nucleic acid code. And what we need to do is shift it into protein code. And proteins are made of amino acids. They are totally different things. So we need to somehow get this to this. And that's what the translation does. It's probably the trickier of the two processes. What also can happen is we can obviously pick control points anywhere throughout this whole thing. Okay, so just as an aside, well, not a total aside, you have to have some idea of this. Human Genome Project. Okay, so 1990 they started out to sequence the entire human genome. Completed it in 2003. So it took 13 years. If you want to sequence an entire genome now, it'll take 1 or 2 days. Not the analysis of it, but the sequence of it. So it shows you just how much the technology has improved in comparison. Now they originally thought, roughly expecting in something as complicated as a human in terms of multicellular organisms, 1 to 200,000 genes reasonably. We now reckon are 22.5. So not a huge amount, really, for how complicated you are. Which causes problems, because we know there's more proteins than that, but we'll come back to that one. So in reality, we're talking about less than 2% of the human genome actually coding for DNA. So all of the proteins that make the organisms work, every single bit of it, less than 2%. So you've got 98% that is not actually making proteins, and you haven't got rid of it. It's hung around for the entire evolutionary life of the genome, so it has to have purpose. Otherwise, you would have got rid of it over time, because what would have happened is you would have had a cell that lost some. it wouldn't have slowed the cell down. In fact, it would have speeded it up because it would take less time to replicate, and so therefore that would probably have given an advantage and it would have continued on. However, we've not done that. So trying to figure out what that non-coding DNA is doing is a huge part of genetic research now. And we've got some stuff we know, some stuff still don't. So the next most important thing in this whole process is a bit better understanding about RNA. Now you have, and I am highlighting this in particular, these are three main forms. There are many different forms of RNA, but there are only three that you're going to need to know for this process. So we have mRNA, messenger. So what that's going to do is to copy a DNA section, so a gene, and then that's going to take it out of the nucleus. Okay, we then have tRNA, which is transfer RNA, and what that's doing is taking amino acids that are floating around in the cytoplasm and taking them to the ribosome to help connect up to make the actual protein. And finally you have rRNA, which is ribosomal RNA, and it's part of the whole ribosome complex. Okay, so to be very clear about how RNA is different from DNA, you are quite often asked to name structural differences. So here we go. First off, the sugar, and it's not a big difference. I showed you it earlier when we were looking at DNA. It is just this. On the ribose in RNA, you still have an oxygen here. The deoxy in DNA has taken away an oxygen. We have nothing here. Now that doesn't look like much, one tiny shift, but this has huge impact on the exact angles you're going to find inside this particular sugar. From a bonding point of view, this actually allows you to have another hydrogen bond. The carbon shape of this sugar is just slightly different. There's a tiny change in the angle. It matters for another structural difference I'm just going to go into. Okay, so we also have the base difference. So your standard kind of nucleotide structure, phosphate, sugar, base, that's still exactly the same. And the phosphate is exactly the same. The sugar is now ribose rather than deoxyribose. And the bases are very similar. We have three that are the same, these three, but what we also have is uracil. Okay, so in this case, we now have base pairing, which is slightly different than you would expect. So G to C is fine, but now we've got an A to U. Okay, so you just need to know that this is a new base and you don't have thymine. So that tiny difference in the sugar drives a massive difference in how it can behave. So here's your DNA double helix over here. Okay, looks like a double helix, and this is the one that you've seen throughout. Okay, it's very, very, very precise. The angle of each sugar to phosphate in here has to be absolutely perfect for it to connect round and do this kind of spiral. In RNA, that sugar is just slightly different from the sugar that you've got in DNA. So instead of being able to keep going and spin into a spiral, this can only hold a kind of straight line, curved line, okay, but single-stranded. So you can get it to kind of link on for long enough to pair up, but as soon as you let it go, it will just peel back off again because it does not want to stay in this helical shape. So what you'll get is a single strand rather than a double. And so those three are your structural differences, and you're expected to be able to give those as structural differences. There is another difference, which is where you're going to find it. So RNA in eukaryotes, you're only going to find your DNA in your membrane-bound organelles, so that's the nucleus and the mitochondria and the chloroplasts. But RNA is going to be everywhere. You're going to get it in the nucleus, where it's busily copying away bits of the nucleus. You're going to get it in the ribosomes, where it's doing the actual translation, and it's going to be floating around the whole of the cell where it's picking up amino acids. So overall, be aware of the differences and make sure you're very careful if they ask you things like give the structure of a nucleotide. If they just say a nucleotide, then that's fine, you just say it's phosphate, sugar and base. If they ask you for it for a DNA nucleotide, you're going to have to tell the difference here. So you're going to have to say that that is deoxyribose. Oh, sorry, ribose sugar. And if you were asked for it in RNA, you have to say it's ribose sugar. Okay, and then your bases, ATGC here and AUGC for your RNA. Okay, so I'll run through the three types. mRNA. So each triplet of bases on an mRNA strand is called a codon, and each codon will code for a specific amino acid. We're going to look at it more precisely a bit later on. Okay, so what we have is, in this example, here's your strand of RNA running along here. For every 3 bases you go along, so in this case GCU, because remember we've got uracil rather than thymine, that codon would equate to alanine. And you have 20 different amino acids which are all coded for by different codons. And all the way along, it'll just keep moving along, every 3 giving you a new amino acid. Each of these amino acids will then link up together to each other at these points in between here, and now you've got a polypeptide. And we have some special codons that say something different. Like this one says stop, and then you're done. trRNA. trRNA is a very complicated looking thing, but you're unlikely to be given it as a very complicated looking thing. So what matters in trRNA, and you have to be able to recognize and talk about, is that you have 2 sites. At the top you have a site for an amino acid. That is the amino acid attachment site. At the bottom you have an anticodon. So the anticodon is opposite to the codon in the mRNA. The rest of the body of this, all this bit down here, and these are just different versions of how to see a tRNA molecule, they are made of RNA and they help hold the whole thing together. But I mean, you could have something that literally looked like this. There's your amino acid at the top, and here's your anticodon down the bottom, and not even bothered to draw all the scribbly stuff inside. Okay, so what this is doing is the actual translation device. This is taking a code which is in nucleic acid code and shifting it into amino acid code. Okay, so rRNA is part of the ribosome structure. So it's a kind of complex system in terms of it looks very complicated. This is a space filling model from a yeast cell, Saccharomyces cerevisiae. But you're more likely just to see it drawn as a big blob with a little blob underneath it. That kind of thing. So don't worry about dealing with it in that kind of level of complexity. What it does is move along the mRNA strand, matching up using tRNAs to make the amino acid code to make the protein structure. And that is a reasonable rundown. You're going to need to know it in more detail of what you're going to need to do gene expression.